Okay, so in the last class we completed our run through the process of respiration, and then we talked about anaerobic respiration. Today we'll be talking about respiratory substrates. So that is chapter 7A5, A2 book or book 2 for Edexcel, and chapter 12 for the Cambridge A-level book, right, by Cambridge Publications. So we have respiratory substrates and respiratory quotients. That's a U. Okay, so we know that primary source of energy for us is glucose. And so when we consume something, right, that has carbohydrates, right, we know that starch or other carbohydrates or sucrose, right, in the sugar that we use, the processed sugar, right, those can all be broken down to glucose. But if there are other forms of carbohydrates you take that have either fructose or galactose, then those are processed to glucose, right, and then used in the process of respiration to produce ATP. But what if glucose is not available? Right, so the secondary source are lipids. And even though they're the secondary source, they actually produce far more energy as compared to, or I should say, release, right? Far more energy as compared to glucose. And that is a reason why in mammals, right, or many animals, not necessarily mammals, lipids are used as an energy reserve. So, right, you have fat deposits in fish, in birds, right, camels, bears, right? So animals that are either in cold temperatures, right, or in situations where they are not able to access food readily, and so fat builds up in their body. And the hump of the camel or fish have a lot of fat in them. Tend to, right? I mean, fat deposits have other uses or benefits such as insulation, but right now we are talking about, right, them as an energy source. And so bears, for example, that hibernate, or other animals that hibernate, they eat a lot before going into hibernation. They build up their fat reserves, and then those are the reserves that are used to provide energy during their state of hibernation, right? Because obviously they're asleep for a long period of time. Unfortunately, that is not what happens in humans, right? So if we build up fat, we have to work really hard to get rid of it. But, right, even in humans, when fat is metabolized, it does release a large amount of energy as compared to glucose. But again, that is a secondary source. Now, how does that work? So, fatty acid molecules are long chain, right, carbon chains or hydrocarbon chains. And as we saw in the process of glycolysis, what happens is glucose is broken down into two, three carbon molecules, and then it's put through the link reaction and the Krebs cycle and so on. And the carbon is removed, and that releases hydrogen, right? And that is basically where all the energy is coming from, the hydrogen that's been released, right? That goes on to the hydrogen acceptors, the NAD and the FAD, and they go on to release their hydrogen into the electron transport chain and oxidative phosphorylation, which results in the production of ATP. And glucose is just 6 molecules, right? So when you have long hydrocarbon chains, you would expect a lot more energy to be produced, and that is exactly what happens. Now, how that happens is, right, a 2 carbon section is removed, right? So 2 carbon sections are removed from the fatty acid molecules, right, as CO2. That is a waste product. Oh, sorry, the CO2 comes later. Right, the 2 carbon sections are removed, right, and form. Just go back on that, right, as acetyl coenzyme A, right, and that enters the Krebs cycle. Also, hydrogens are removed along with the carbons, and they are picked up by the right hydrogen acceptors. And they go on to the electron transport chain. Now, right, this one actually happens much faster than this, right? So acetyl coenzyme A is fed into the Krebs cycle, but comparative to how quickly that happens, the hydrogen removed is picked up by the hydrogen receptors, and they move on to the electron transport chain much more quickly as compared to acetyl coenzyme A going into the Krebs cycle. And right, as just to give an example or a sample of how much energy can be produced, stearic acid is a fatty acid molecule that is naturally present in mammals, right? Animals and plants as well, right? Some cases. It is an 18 carbon molecule. And a single molecule can produce up to 180 ATP molecules, right? As compared to one molecule of glucose producing 30, right? So, and right, so I mean, it's 6 times the amount. Obviously, you have a longer carbon chain, and that is the whole reason. But molecule to molecule comparison, fats produce a whole lot more of energy. When you talk about lipids, it's not just fatty acids, it's also glycerol, right? And glycerol is also used. So what happens is glycerol is phosphorylated, and converted into something called GALP. Now that's what's given in the book. what this is, right, is a abbreviation of its chemical name, which is glyceraldehyde 3-phosphate. And it's also known as G3P or GLP or other names, right? But GALP is one of them, right? And that's what's given in the syllabus. And this GALP, right, enters glycolysis, right? So the very first step of respiration. So that is used. And just to tell you where it makes an entry, if we just skim back, go a bit further back, but it's still right. I mentioned that the phosphorylated 6-carbon sugar, right, is the fructose 1,6-base phosphate. Then you have the 2,3-carbon sugars, right? The triose phosphate, as it is called in the Cambridge, right? So it's not mentioned here in your Edexcel book, but it is mentioned as triose phosphate in Cambridge. This triose phosphate is gamma, right? So this is where glycerol enters after being converted to GLP by phosphorylation, right? And so the carbons from the fatty acids enter the Krebs cycle. The glycerol enters the process of glycolysis, right, as the 3-carbon phosphorylated sugar molecules. And then obviously proceed through the remaining process of glycolysis, link reaction, Krebs cycle, and finally oxidative phosphorylation electron transport chain. Now proteins. We also have proteins, but proteins are a last resort. By the way, any questions? All good?
Not. Yes.
Proteins. So can proteins be used as a source of energy by the body? Well, the short answer is yes. But again, it is as a last resort. And that is because proteins run the show in the body. Right? Proteins are that important. You've got all sorts of enzymes, hormones, right? Your muscles, your signaling molecules other than enzymes and hormones, right? So neurotransmitters and stuff going on in the cells, so microtubules, right? The cytoskeleton of the cell, and the proteins in the membranes and XYZ. I mean, they're everywhere, right? Your skin, your hair, your nails. I mean, right? Keratin and whatnot, right? They're all protein. They're all protein, right? So protein is an energy source of last resort. The body only turns to it when it feels that it is starving. So it does produce, right, energy. It produces about as much energy as carbohydrates. But in case the body senses it's starving, so right, it starts breaking down muscle mass. And this is where, if you're dieting or trying to lose weight and you're not careful about how you're doing it, when you stand on the weighing scale and you see that you've lost some of your weight, you have, but it's not the healthy weight loss, because it's possible that you're doing it wrong and the body feels that it's starving and it starts breaking down your muscles. And that's why when you're dieting, you might feel weaker. And it's not just like, you have a glass of juice or something to eat and you feel weaker generally, right? You get tired more quickly, and that's because you're losing muscle mass. Now, right, proteins cannot be used directly. So what they are is that, they're broken up into amino acids, and then those amino acids are deaminated in the liver, and that means that their nitrogen group is removed, right? Or ammonia is removed. The ammonia is obviously processed and removed as a waste product, converted to urea, etc., etc. And then what's left after deamination is converted to carbohydrate and then used as energy. Now, if I just take you to the book, they have a nice chart showing the interconnection between all of this. Let's put that on screen. There. So all the different respiratory substrates and where they enter the respiratory pathway to provide ATP. So you've got starch or glycogen. Glycogen is the initial store, right, in the liver. Starch is something that we consume. This is the process of glycolysis and the Krebs cycle. You've got fatty acids. They can be broken down to acetyl coenzyme A or converted to fat, or they could be broken down to glycerol, which is the phosphorylated 3-carbon sugar entering glycolysis. Right? Amino acids are obtained from proteins, and they can be broken down and converted to acetyl coenzyme A or pyruvate, right? And then they can enter, right, the Krebs cycle. And obviously, right, between each of these arrows are multiple steps of the amino acids. It doesn't mention here, right, the deamination and conversion to carbohydrate, and then pyruvate, and then entering, right, or entering as acetyl coenzyme. But this is a nice chart that summarizes the pathways of different respiratory substrates. Any questions?
No, sir.
Okay. So coming back to the whiteboard. Now that's all there is to talk about as far as the respiratory substrates are concerned. So next, we will talk about respiratory quotients. Now, what is a respiratory quotient? Right, it is a measure of how much oxygen is utilized. So, how much oxygen is utilized and carbon dioxide released carbon dioxide released, right, when respiring. And so the value, because it has been studied, can tell us which substrate is being respired. and which means which substrate is being used by the body to provide energy. And there's a formula for this, which is pretty simple. So the respiratory quotient, right, is also given as RQ. So you can write that. So RQ equals to carbon dioxide produced over oxygen used. Okay. Now, how do we get this? Well, we used balanced equations, right, of the respiration of each of these substrates. So we know that we have glucose, right, and the balanced equation for that is so C6H12O6 plus 6O2 will give you 6CO2 and 6H2. And so you can see that the number is the same. And so RQ for glucose, right, or carbohydrates perhaps in general. So let's write that over here. Equals to 6/6, which is 1. And 1 is the maximum RQ, right, for aerobic respiration. Right? It can be 1, it can be less than 1, it can't be more than 1. Okay? For aerobic respiration, which means oxygen is being used. If we talk about lipids, the RQ is about 0.7. Right, so an example, right, in this case is the fatty acid called oleic acid, right, from olive oil. With the stylus. And the balanced equation for its respiration is C18H34. Sorry about that. C18H34O2 plus 25.5 O2. So yes, when balancing, right, we can use decimal points. And that will give us 18 molecules of carbon dioxide. Now that is pretty much as we need to know, but still to balance it out, 17 molecules of water. And so, right, can I squeeze it in here? RQ, right, is 18 divided by 25. And that gives us 0.7. All right. And just to mention this on the side, the RQ. So in fact, no, I have to talk about anaerobic respiration. So we'll take a new page. I don't get. I mean, I have an infinite number of pages, but I'm still trying to squeeze it in. So proteins. Now, just to give you that, the RQ for proteins is 0.9, so very close to carbohydrates. Now, what happens in anaerobic conditions? Right, well, in anaerobic conditions, there is no CO2, no O2, sorry, lots of CO2, no O2. Right, and so if we take the example of, the fermentation of glucose and yeast that we did, so glucose in yeast. So you've got C6H12O6, no oxygen is being used, and you are getting, right, two molecules of lactic acid, carbon dioxide released, and energy. Right, so two molecules of C2H5OH. Oh, sorry, this is ethanol. Lactic acid is in muscle, so this is ethanol. Plus two molecules of carbon dioxide and energy. And when you go on to take the RQ, you have carbon dioxide, which is 2, divided by 0, which is no O2, and you can either write it as infinity or as, right, we were taught undefined, which basically means that, right, if your RQ is, as I mentioned, right, for aerobic respiration, it cannot be greater than 1. If your RQ is greater than 1, that means there is some anaerobic respiration going on. How much depends, but anaerobic respiration is going on. Right? It might be completely anaerobic, it might be a mix, it depends. Right? Also, although I just talked about anaerobic respiration, but right, if the RQ, right, so if this is being studied for a certain organism, right, if the RQ comes as 1, that means carbohydrates are being used for respiration, right, as the primary source. If it is less than 1, that means there is a mix of carbohydrates and lipids. Again, right, you're not considering proteins because proteins is last resort in emergency when the body is just focused on surviving, right? If it feels threatened and needs to survive, right? So that is something to keep in mind. And we are done with right, chapter 7a. Okay, now that we're done with respiration, the next chapter that we're moving on to, because we have time and we still have spirit, is chapter 5a, photosynthesis. Again, the reason we're jumping about is because we are following the syllabus pattern that's given in the Cambridge specification, just so that we can align the topics, right? The content is a bit different here and there, so that will be highlighted if required. So chapter 5a is photosynthesis. Section 1 of chapter 5a is about the importance of ATP. Now, we've just come out of the chapter for respiration, and so I'm sure we very well know what the importance of ATP is. So I'm not going to go into the discussion of the molecule of ATP and how it's formed and it provides energy, etc., etc. It's very briefly given because the details obviously come later, which you've already covered. There are one or two points mentioned in this chapter, however, that I'd like to mention in passing, and then move on to section 2. So section 1, the importance of ATP. Now, what they mention over here, right, are the terms used to classify organisms based on whether they're able to make their own source of food or not, right? And they also mention that the sun is the primary source of energy, right, for all life. So sun is the primary source energy for all life on Earth. So we talk about plants use energy to make up their molecules, and we get that it's stored as chemical energy, and then we eat the plants or we eat the animals that eat the plants, etc. And so the question is, where is that energy coming from? It's coming from the sun, right? And from there, the plants use the sun's energy, and more specifically, right, it's light, because the sun is also providing us with heat, and convert that to chemical energy, right, in organic molecules. And because they can do that, plants are called, they're called autotrophs. Right? Auto meaning self. Right? So they can produce their own food source. And then all the other organisms that either feed on plants or, right, feed on the animals that feed on plants. Basically means, right, that they cannot, right, produce their own food. Or in other words, they cannot utilize the energy from the sun. In that regard. And so these are called heterotrophs. So they're dependent on other organisms for their energy source. All right. Then, right, while discussing ATP, they highlight the fact that, right, when bonds are made, energy is used, right? And that makes sense, right? I'll say when bonds are broken, energy is released. right? And we've seen the breakdown of glucose and the fact that it has a, right, so respiration has a negative, right, delta H, which means energy is being released. Photosynthesis is the process where uh, glucose is being made, right? So bonds are made. And so we'll see that it has a positive delta H, means energy is being used, right? And that energy, as we just talked about, is coming from the sun. Okay, anything else worth mentioning? Not really, no. All right, they mentioned the electron transport chain, they mentioned redox reactions, we've done that. They mentioned ATP being catalyzed and a phosphate bond being broken to convert it to ADP, adenosine diphosphate, and the enzyme, right, that is doing that, that's catalyzing the reaction is ATPase. Uh, right, so I'm putting an asterisk not because it's out of syllabus, right? So ATP, when it is, right, converted to ADP and phosphate is released, this is called hydrolysis. This is a reversible reaction. And the reverse, in general, when bonds are being made, so when bonds are being broken, it's called hydrolysis, right? And in general, when bonds are being made, it's called condensation. In this particular case, it can also be called phosphorylation, as we did mention, right? And both of these processes, so both, the reverse reactions are catalyzed by the enzyme ATPase or ATP synthase, whatever you want to call it, right? Okay, so I'll just write bond breaking is hydrolysis, and bond making is condensation. And yeah, so we are done with section A. Now, I'll just briefly introduce the topic of section 2, so 5A2, where we're going to discuss chloroplasts and chlorophyll. So 5A2, chloroplasts and chlorophyll. And this is where, right, first of all, we are introduced, in A level, right, we already have the introduction from our IGCSE or O level to the summarized process of photosynthesis, just like we have the summarized process of respiration. So in this case, we have carbon dioxide, so synthesis, right, is making glucose from carbon dioxide and water using light energy from the sun. So we have CO, well, let's write the word equation first. Carbon dioxide and water. We put light, and they also mention chlorophyll. chlorophyll, and that's giving you glucose plus oxygen. And in this case, oxygen is a waste material. So the balanced equation for that is 6CO2. It's pretty much the reverse of respiration. 6CO2 plus 6H2O. Right, again the light and the chlorophyll, and you're getting glucose and 6 molecules of oxygen, and you have a delta H of about 3000 kilojoules, right? Energy that is being used, 2880 kilojoules of energy is being used to make glucose. All right. Now I said that in essence, if I don't need to write that. So in essence, when you look at the equation for photosynthesis, you realize, oh, well, this is pretty much the opposite of respiration. And yes, it is. But, right, we also need to understand, right, the students tend to confuse that, oh, well, respiration happens in animals and photosynthesis happens in plants. No, right? So, animals, respiration only, if you're talking about these two processes. Right? Whereas in plants, you have respiration and photosynthesis happening. Right? And that's because plants are living creatures, they're living organisms. So obviously they need to respire and they need to make that ATP and then use that ATP for all of their functions, just like animals need to do as well, right? Or microorganisms respire, all of that. So it's right, just that photosynthesis is, you know, dominant or prevalent or, happening during the day. So is respiration, right? Because the plant is alive, obviously. And right at night, photosynthesis slows down to a crawl. It doesn't stop, right? It doesn't go to zero. It just slows down to a crawl. And right, respiration is the major chemical reaction happening in the cells, uh, when we compare these two. Also, right, to understand, to realize that both of these are happening in plants at the same time, the oxygen and the carbon, the glucose that is produced as a result of photosynthesis, right? Some of it is released, right? But some of it is utilized within the plant. It's not released, right? It is utilized for respiration, right? And the glucose as well. Some of it is utilized for respiration, then and there by the plant cells, and then some of it is taken, it is stored or converted, and we get into details of this, right, to amino acids or lipids or whatever, right? Amino lipids, amino acids, and lipids, and other, right, whatever biological molecules are required. So that tells us yes, yes, right? So just don't have that notion plants don't respire. They do. And I will stop here, right? So uh, tomorrow, right, we will go into detail about chloroplasts and chlorophyll, and then talk about photosynthesis, which also occurs in a number of different steps. So what we have is the summarized equation, right, as we should understand by now, right? It's not that simple. Any questions?
No.
All good?
Yes.
All right, then I will see you tomorrow.
Inshallah.
Sure. Thank you.
Thank you. Welcome, sir. I mean, yes. Allah Hafiz.
Allah Hafiz.